orient you again in the schedule. Right now we're here, week 12, so we basically just have today's lecture and one more lecture to cover the material that will be on the next exam, which is a week from Thursday. So Tuesday we'll have discussion and review session on Tuesday. So last lecture we talked about stem cells and introduced that stem cells by definition can self-renew and differentiate, and that cell differentiation is controlled by regulation of gene expression. And we talked about how gene expression is regulated at the transcriptional level as well as the translational level. And we talked about how physical structure of the chromosome can affect gene expression. So we talked about heterochromatin versus euchromatin, and we talked about chemical modifications of histones and DNA, like we talked about in discussion today. And then we introduced the idea of the local environment, or the stem cell niche, that controls whether a stem cell is going to self-renew or if it will differentiate, and what kind of cells it will differentiate into. And we ended with the example of hematopoietic stem cells as an example of a type of stem cell. And so we talked about its stem cell niche as being the osteoblast cell, or the bone building cell, that interacts with the hematopoietic stem cell to help it continue to self-renew. And that certain signaling pathways such as Notch and Wnt are important for interactions between the hematopoietic stem cell and the osteoblast, as well as expression of adhesion molecules such as N-cadherin. And we talked about how, depending on the growth factors that are present in culture, a hematopoietic stem cell can be induced to differentiate into different cell types, like a granulocyte or a monocyte. And as they differentiate, these hematopoietic stem cells will change the genes that are expressed, and this results in different gene transcription networks, as well as expression of different genes or specific genes at different times. And so we talked about hemoglobin as an example of this, how in early embryogenesis there's one type of hemoglobin expressed in red blood cells that has higher affinity for oxygen than the one that is expressed in the adult hematopoietic cells. Okay, so kind of incorporating all of the concepts of developmental biology that we've discussed so far. So today we're going to pick up where we left off, and we'll talk about muscle cells as an example of another stem cell type. And muscle cell stem cells are increasingly being investigated research-wide. So just to review, going back to the somite, right? So we've learned that somites develop into the muscles and skeleton of the vertebrae, and that as they are patterned, there are sections of the somites that will become dermomyotome, and the dermomyotome contains the myotome, which is what forms the muscle cells, and that's what we're going to pick up from today. Okay, so when a muscle cell differentiates, like hematopoietic stem cells, there are several stages of differentiation. And so this is the neural tube, this is the notochord, and these are the specific, myotome cells that are present in the somite. So first, there are growth factors that are secreted from the neural tube and the notochord that signal to these myotome cells that they are going to be myotome cells. Right? And so that's determination. And some of the signaling pathways that are involved in this include Wnt and hedgehog. And there's expression of specific transcription factors in the myotome cells, such as MyoD and MyF5. So So these are muscle cell specific transcription factors. So they were only expressed in cells that are pre-muscle or will become, or that are destined to become a muscle. So the myotome cells will differentiate, they change their shape, and they also rapidly divide. And when they're dividing, they're called blasts or myoblasts. And fibroblast growth factor is important in increasing their cell numbers. And then as you have more cells, and with increasing age, the myoblasts will align themselves. Multiplication will stop. They interact with each other as well as with extracellular matrix proteins such as fibronectin. And they utilize integrins and tempirid adhesion molecules to produce these aligned strips of cells. And then after they've aligned, the muscle cells will form what are called myotubes. So muscle cells are unique in that they actually fuse with one another. And so your muscle fibers basically consist of a bunch of fused muscle cells. And this is important for the electrical transduction that they get when they get the electrical signals from synapses. So they can travel faster because there's only one cell membrane. So you get initiation of myotube formation, and then you get mature myotubes. So here you see there are more cells fusing together. And eventually you have a true muscle fiber. And then the muscle stem cells are found to be located adjacent to these muscle fibers and in direct contact with them. And at this stage, especially if it's, for example, a cardiac muscle, you'll start to see normal contractions. Okay? Maybe some more here? Are you okay? Raise your hand if you need help. You need medical help. Okay. Okay, so this is a summary of what I just told you. So again, we start with dividing myoblasts. So these can be considered muscle stem cells. They align, they fuse, and they form a muscle fiber. Okay, and this shows what's occurring at the molecular level as these mesodermal progenitors become muscle fibers. You have external signals that promote genes that are important for determination to the muscle fate. So again, here we see MyoD and My5 transcription factors being activated. And then you get differentiation into more mature myotubes and maturation into the final fiber. So there's a lot of muscle-specific genes that get activated sequentially as the muscle cells mature into fibers. Okay, so this is a picture of a muscle fiber. It's kind of shown here in the red in the background. And here, this cell is what's called a satellite cell. And the satellite cell is a muscle stem cell, and it is an adult type stem cell. So it's here you see it's situated right on top of the muscle fiber, and there's usually some cells adjacent to it that accompany it. So satellite cells are activated to divide in the case when the muscle is damaged, or like when you exercise and you kind of slough off some of your